Good evening, class. Sorry for the late class today. It's just 10 minutes late. I was just, I just came from work today. I was just not feeling well. I slept for 5 minutes and I just woke up at 7 and I totally forgot that I have to take your classes today. But somehow I'm awake and good to go. And so otherwise, we might have uh, missed our class. So, Abhay Shrivastava, does sarcolemma play any role in myocardial infarction? Yeah, why not? Sarcolemma is the thing playing role with the contraction of your cardiomyocytes. If they are not working well, it will lead to the myocardial infarction. There are many papers related to that. So, so not without wasting our time, we should start. Not here, here. Some days are just tiring. So, and students, those who have not subscribed to my channel, please do subscribe. Only 34 students are present today. Very few people are being joining. It's not good. Anyhow, those who are joining, they want to learn something. So keep going good. Bioinformatics and PPT. So DNA fingerprinting. So where are the other students actually? As the prolonged death of cells, cells are not giving as good they should work. There's a lot of signaling pathways are not working as they should. There might be a lot of phosphatases are present, leading to a lot of calcium deposits. There are many reasons for the same. There are many reasons. So let's start with DNA fingerprinting. This is done, basically DNA fingerprinting is done to check to check the DNA fragments of the crime scene and the person who did that crime, whether they belong together. That's why we do it. So why use DNA fingerprinting? So DNA fingerprinting is a way of telling individual of same species apart. So DNA sequences are a variable and can therefore be used as identifying characteristics. So DNA fingerprinting has advantages over others source of evidence, fingerprints, blood type, etc. And is highly accurate and can be gathered from trace crime seen evidence. So one way of restriction fragment length polymorphism is possible. So in this, each enzyme is cut at very specific DNA sequences, and then we check with the help of gel electrophoresis. So here you can see BAM H1 is cutting our DNA sequence from GG part and then giving rise to sticky ends. So individual 1 giving rise to 5 DNA fragments. And individual 2 cutting from 3 giving rise to 4 different segments. So individual 1, individual 2. So that's the main summary of this. Essentially, once the DNA has been cut, then we can check the various sizes of DNA fragments, and each individual binding pattern should be different because restriction enzymes will cut each other person's DNA at different points, and fragments of different sizes will travel different distances along a gel when electric current is passed through it. So here is your across gel electrophoresis. So different binding patterns from different individuals. So you have this one double here, double, but each one showing different sizes. So that means they are from different personalities. So how do you analyze them in the lab? So first photography of fingerprinting was done in 1985. So DNA fragments show different patterns from one person to the next, and this is because of the it could be used for the paternity disputes and as forensic evidence. So specimen collections could be lick envelope, dirty laundry, cigarette butt, saliva. These all specimens could be used. Uh, Animals of evidence that could be harmful for evidence could be sunlight, high temperature, bacteria, moisture. And it's ideal to have 1 ml of fresh whole blood treated with EDTA. So the same principle RFLP, individual 1 and 2, cutting out different perspective, giving rise different segments and different results. It's quite sad. There are 170 students, and so far 50 students have joined so far. And other students are just sleeping and they don't want to be part of it. So that means this training doesn't have much meaning down there. It's just done for the sake of certificates for all other 100 students. So for 58 here, I highly appreciate you, give you highly respect. And for all other students, I think it's a business. And which is not appreciable at all. You paid some fees that you might get a certificate from IIT, but believe me, without attending that, that will be not worth it ethically for you. But rest, you know the best. So then there is southern plotting to check the agarose gel with a probe. You switch to the nylon membrane, and then membrane with DNA fragments are transferred to it. Then you can see the in the southern blot the bands that were present in it, and this is not required to go for variable number tandem repeats. This is means a particular sequence is present in multiple times, like GATC, GATC. It's present 5 times, 2 times. Then VNTRs usually occurs in introns and can be amplified by PCRs and run on agarose gels to produce unique DNA fragments. So polymerase chain reaction. So lab technique used to amplify segments of DNA. So reaction requirements. So for the PCR, as we have discussed before, you need template DNA, DNA primers, DNA polymerase, and nucleotides. And then the PCR machines, which are placed in a machines called thermocycler, and machine cycles through 3 temperatures in this. First at 94 degrees Celsius, then 50 for annealing, then at 72 for elongation. And then it is repeated for 30 times. Then you have double, like millions of base pairs at the end. What are the applications of DNA fingerprinting? It helps for the diagnosing diseases, paternity testing, forensic field, victim crime scene suspicion, genealogy animations. Yeah, that's it. So that was the very short and brief about the application of DNA fingerprinting. So So something about now restriction enzymes. So restriction enzymes, the first is the introduction part with the mode of action, star activity, nomenclature of restriction enzymes, type of restriction enzymes, DNA ligase, kinase, reverse transcriptase, clinofragments. So all these parts belong to this. So in the introduction part, so it is the ability to manipulate DNA in vitro depends entirely on the availability of purified enzymes that can cleave, modify, and join the DNA molecule in specific ways. So basically, they play quite important role in manipulating the DNA, and they have vital role to play in process of genetic engineering. So various enzymes used in genetic engineering are as follows: that is, nucleases, restriction enzymes, DNA ligase, kinases, phosphatases, self-transcriptase, terminal deoxynucleotide transferase. So nucleases. So nucleases are a group of enzymes which cleave or cut the genetic material. That is like DNA and RNA. So in the DNases, so there are two types, DNases and RNases. So nucleases are further classified into two types based upon the substrate on which they act. So nucleases which act on cut DNA are classified as DNases, whereas those which act on RNA are called RNases. So DNases are further classified into two types based upon the positions. So DNases that act on the ends or terminal regions of DNA are called as exonucleases, and those act as the non-specific regions, the center of DNA are called endonucleases. And exonucleases require DNA strands at least 5' and 3' end. Further, restriction enzymes, DNases act on specific positions of sequence of DNA that are restriction endonucleases. And further, how they work. So basically, the working of restriction enzyme is to recognize the reason of methylation, that is presence of methyl group on the DNA at specific nucleotide. If there is a methylation in the recognition sequence, then it is just false of the DNA and does not cut there. If only one strand the DNA molecule is methylated, other strand is not methylated, then restriction enzyme will methylate and other strands are the required positions. The methyl group is taken by the restriction enzymes for as adenosylmethionine by using modification sites present in the restriction enzymes. However, type 2 restriction enzymes takes place with the help of another enzymes called methylase and methylate the DNA. There is also a thing called star activity. So sometimes restriction enzymes recognize the sites, cleave the DNA strands at the recognition site with the asymmetrical palindromic sequence. For example, BAM-H1 cuts a sequence at GATCC, but under extreme conditions due to the low ionic strength, it will cleave with any of the following sequence like NGATCC, GPOTCC, and such activity of restriction enzyme is called star activity. Okay. Yeah, I'm continuing students, but it's hard actually if others are not attending. Okay, all is good, all is good. Then the nomenclature of section enzymes. Sorry if my voice is low today. It's just not feeling so energetic at the moment, but I'm trying my best to be loud as enough. So, as large number of friction enzymes have been discovered, so there's some how to give them names so that we can avoid the confusion. So, Smith and Natings in 1973, they created restriction enzymes would have specific names which would identify it uniquely. The first three letters in italics indicate the biological source of enzymes. The first three letters being the initials of the genus and second and third being the first two letters of specific species name. So thus restriction enzymes from Escherichia coli called ECO, Haemophilus influenza becomes HIN. That is E. coli, you take first word from the genus and the two words from the species. From hemo, H and then IN from there. Diplococcus, pneumonia, DPN and so on. Then comes a letter that identifies the strains of bacteria, EcoR strain. So finally there's a Roman numeral that is EcoR1, that is it's belong to the first enzyme, and the for second EcoR2. So like this. So there are types of restriction enzymes, that is type 1, 2 and 3. So type 1 and 3 have an ATP dependent restriction activity and modification activity resident in the multimeric proteins. So both these types recognize unmethylated recognition sequence in DNA, and type 1 enzymes cleave the DNA at random sites, whereas type 3 cleave at specific sites. So type 2 restriction modification system possesses separate enzymes for endonucleases and methylase activity and are the most widely used for genetic manipulation. So type 1, it's cleave the DNA somewhere between 400 base pairs to 10,000 base pairs or 10 kilo base pairs right or left. So cleavage site is not specific and they use the enzyme requires magnesium plus ATP and acetoadenylyl methionine for cleavage or enzymatic hydrolysis of DNA. So, and there's a type 2 section enzyme. This one recognizes sites of the DNA within the recognition site of sequence, and they requires magnesium ions and cofactor for cleavage activity and can generate phosphate and hydroxide. So enzymes of this type are highly important because of their specificity, and type 2 restriction enzymes are further divided into two types based upon their mode of cutting. And then there's a type 2 restriction enzymes, blend and cutters. So blend and cutters type 2 restriction enzymes of this class cut the DNA strands at same points on the both strands of DNA within the recognition sequence. And the type 2 restriction enzymes cohesive end, this is a blend and cutters and the cohesive end cutters. Cohesive end cutters type 2 restriction enzymes of the class cut the DNA strand at different points on the both strands of DNA. So type 3 restriction enzymes of this type recognize the recognition site but cut DNA 1 kilo base pairs away from the restriction site. So these enzymes are made up of two peptides and require ATP, magnesium ions and S-adenosyl methionine for action. So this is the all the differences between type 1, 2 and 3 with respective of the structure, compositions, cofactors, recognition sites and cleavage. So some restriction endonucleases and their recognition sites. So these are enzymes like MBO, MCP, ALU, HAE, with the recognition site as a GATC, CGG, AG, GG, AC. And there are six base cutters cutting from A from GAT, CT, AT, CGAT, CAG, CTG, and eight base cutters cutting the eight base pairs like this, not LNS, BFL. So you should be remembering these recognition sites here, which are the one, and also the enzyme names, with the four base cutters, six base cutters, and eight base cutters. Then there's a enzymes for restriction enzymes. After the restriction enzymes comes a DNA ligase, which helps to join two different DNA fragments. And in this, the cohesive ends generated by restriction enzymes will anneal themselves, performing of the hydrogen bonds. But sometimes it's also joined with the help of covalent bond by catalyzing the formation of phosphodiester bond between adjacent nucleotides. So DNA ligase isolated from E. coli and T4 bacteriophage is widely used. So T4 ligase requires ATP as a cofactor and E. coli ligase requires NADP as the cofactor. The cofactor is first split ATP minus AMP plus 2PI, and the kinase is the group of enzymes which adds a free pyrophosphate to widely variety of substrates like proteins, DNA and RNA. And in kinases, it uses ATP as a cofactor and adds phosphate by breaking the ATP into ADP and pyrophosphate. And they are being widely used in the molecular biology and genetic engineering to add radio level phosphates. Then comes the phosphatases. There are three types: alkaline phosphatases. They are the group of enzymes which remove the phosphate from the variety of substrates like DNA, RNA, protein. They act in the basic buffers such as pH 18 or 9 called alkaline phosphatases. And most commonly alkaline phosphatases are bacterial alkaline phosphatases, that is BAP, and calf intestine alkaline phosphatases, that is CIP. And alkaline phosphatase is a metalloenzymes and has zinc ions in them. So BAP has a dimer containing zinc ions in which, and they are essential for enzymatic activity. They are quite stable and is not inactivated by heat and detergent. Whereas a calf intestine alkaline phosphatase is also a dimer and it requires zinc ions and magnesium ions for action. And CIP is inactivated by heating at 700 degrees Celsius for 20 minutes or in the presence of 10 millimolar EGTA. Then reverse transcriptase. So this enzyme uses an RNA molecule as a template and synthesize a DNA strand complementary to the RNA molecule, and these enzymes are used to synthesize DNA from RNA. And the reverse transcription enzyme is also called as RNA dependent DNA polymerase. Then comes your terminal deoxynucleotide transferase. They are the polymerase which adds nucleotides at 3' end but does not require any complementary sequence and does not copy any DNA sequence. Then RNase-P, it specifically cleaves at 5' end of RNA. It complex enzyme consisting of small protein and 377 nucleotide RNA molecule. And it has been observed that RNA molecule possesses at least part of the enzymatic activity of the complex. Hence, it is an example of ribozyme. Then last and not the least, that is your clean-out fragments. So in E. coli DNA polymerase, one, they consist of a single polypeptide chain, and two fragments are obtained from the DNA polymerase. One is treated with a trypsin, and the large fragment is called as clinofragment. So these fragments is of around 602 amino acids in length, and the functions of clinofragment is to add nucleotides to 3' end and 3 end for 3 to 5 exonuclease activity. So clinofragments add nucleotides by using complementary strand as reference. So if a nucleotide is added by mistake, the base pair is wrong, then by 3 to 5 exonuclease activity could be mispaired. So that was all about reaction enzymes. It was quite texty theory, lots of theory in this, but it's good for your net or any other exam to go for. I'm Sampada. I'm cheered up. I'm cheered up. It's just I have given my whole energy in the morning at the workplace, and I'm okay. It's type one cut the methylated DNA, Prashant. All is good. All is good. All is well. Like in the movie we say Three Idiots. All is well. All good. All good. Let me check the another. So, student, this is how your college internal is going on. So, we are giving students number for attendance, discipline, total, and midterm, and we have to grade for all respective subjects. And your future will be decided somehow for each percentage how things goes on. It's it's a lot of work there down there. So we are done with the part of our 4th day lectures. Actually, it's including your RFLP, DNA fingerprinting, and section enzymes. These 3 lectures. Let's start with this one. This is quite interesting. You all will like it. Oh, the battery is 28%. Just let me get adapter. So next is your protein isolations, quantifications, and purification. So this is all part. It's actually proteomics part, somehow related to your molecular biology, with the how it is homogenized, how it is fractionated, centrifugated, then quantitation, then chromatography and electrophoresis. So this part is also taught during your college days with the biophysical techniques and analytical techniques, and also related to your uh, chromatography, electrophoresis, and all these parts. So first, some people who won the Nobel Prize winner on the chromatography and electrophoresis. So Archer John Porter Martin in 1952 for the invention of partition chromatography, and the same Richard Lawrence Millington Singh in 1952 was the same. And then in Arle William Corin Tislas in 1948 in Uppsala, they found in Sweden 1902-1971 for electrophoresis and absorption analysis. Then John B. F. B. Fenn and Virginia Commonwealth University Richmond uh, from USA with the development of methods for the identification, structure analysis of biological macromolecules. Then for the Kochian Tanaka for the mass spectrometry, and Theodor Swidberg for his work on the dispersed system. For the organelle fractionations with the help of centrifugations to have various structural organs for the discovery. Albert Claude, Christian de Duveau, and George Pledig. So they all shared the Nobel Prize winner in their respective years. Then on the protein sequences, Frederick Sanger in 1958 and 1980 from the University of Cambridge, United Kingdom, they determined the respective results. So this is not from here. Basically, your lecture starts. That is your protein isolation. That is first step to isolate your protein. You have to homogenize it. Yeah, you have to first step to disrupt your cells, then fractionate them with the crude separations of homogenates into fractionation. Then homogenizations will include your Towns homogenizer, sonication, osmotic shock, and French press. Then comes the centrifuge at 500 G for 10 minutes. The homogenate forms the supranatant. Then palette nuclear fractionation is done. Then after the homogenizations, so Downs homogenizer, sonication, osmotic shocks, and French press. Then the fractionations, fractional precipitations, pH heat, ammonium sulfate precipitations, differential centrifugations are done. So centrifuge at 500 G for 10 minutes. You have a supranatant, and then in the initial case you have palette nuclear fractionation is getted. Then if you do it for 10,000 G for 20 minutes, then you get palette for mitochondrial fractionation. Then you go for another 1 hour at 10. Oh my god, my laptop is giving me shock. 100,000 G, it's give cytosol, the soluble proteins with the microsomal fractionation. Then after the uh, homogenization and fractionation comes the centrifugations to separate different particles according to their density. So there are two types, that is differential centrifugation and rate zonal centrifugation. So in the differential centrifugations, you have more dense particles and less dense particles. Yeah. So we centrifuge them and particles settle according to the mass. So heavier will go down, light will be up. And then you stop the centrifuge. And what will you have? You have decant liquid into container. You have to move this the upper part in the bottom, and then we will see the supernatant having different organelles and the palette will have different organelles. In the red zonal, it's basically depend upon the different density actually. So your according to the centrifugal force, particle will settle according to the mass, and here you can see that some particle will settle here and there. Then we keep removing them according to their density, and you have increasing mass of particles being separated like that. So protein isolation, so in centrifugation, you have 3000 G of for 5 minutes you do it. Then pellets with the large organelles are removed for chloroplast and nuclei, and at 10,000 G for 10 minutes. Pellets like mitochondria and inclusion bodies. And then at 40,000 G for 30 minutes, your membrane, plasma membrane, tonoplast membrane, microsomes, secretory vesicles, peroxisomes are being separated out. Then at 200,000 G for 16 hours, pellets like large protein complexes, ribosomes, PDH, protein complexes are being separated out. So, and at the bottom, so far, I will not go in detail about the Svedberg coefficients, but you have to remember that one Svedberg coefficient is equal to 10 raised to the power minus 13 seconds. So this has been coming up in net exams quite a number of times. And S equals to M1 minus mu uh, lambda by F is equal to U2 W square X. Then comes the sedimentation velocity technique here. So it's being showing on the first part, A part, that one solution has low density solution, on the right is the high density solution. Then you centrifuge tube, density gradient is done. Then you layer the samples and then separations of sedimentations coefficients. Then you have all fractionations collected through over the bottom of the tube. So these are the various Svedberg coefficient values for different molecular weights. And this is the sedimentation velocity technique. So on the x-axis is the sedimentation coefficient S, and on the y-axis is the density G centimeter minus 3. So what you can see is that there is microsomes are being separated, most viruses, ribosomes, DNA, RNA. So all have different values, sedimentation values, and for different density to be get separated out. Now, once we have separated out our protein that you're looking for, your organelles that you're looking for, the DNA and RNA you are looking for, with the help of a centrifugation technique, and we also have homogenized and fractionated in the first step. Now comes how much protein did you have at the end? You know, how much? Oh my God, sorry, my laptop is giving big shocks today. I have to keep regular check if everything is fine here. Shushan, what is ISSR mark? I never heard them, sorry. I have to Google them. Interspecific, yeah, it's they are being repeated. They're using for the PCR for the microsatellites. Okay. Interspecific simple intersimple sequence repeats. So for the PCRs and all these things, they are done. Okay, so now, oopsie, we are done with this lecture. Yeah, so to check your protein, so we do this Bradford assay with the Lowry method with interference. So Comosi blue G250 dye appears to bind most readily to adenines and lysyl residues, and this specificity can lead to variation in the response of the assay to different proteins, which is the main disadvantage of the method. So this is your Bradford assay.
In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled Sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not hit cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
So that was about Bradford assay. And then this is a protein quantitations, other technique being produced in biotechniques paper, saying that adding dye to sample, heat sample for 10 minutes, cool to room temperature, then you read fluorescence out of it. Then ultrasensitive for protein based. In this, at then you have one on the y-axis your fluorescence, on the axis of protein microgram per ml. Then you create this chart, and then you can calculate according to the fluorescence how much protein is your present in your sample. So this part, so we are done with the quantitation part today, and we will start with the protein purification part tomorrow. So I will share this file with you. It's a big file, 336 MB. So it will take some time to go overload. So you will have no next class. The time is quite limited. So we will start from tomorrow, um, and continue with it with the protein purifications. So we're done with the today we are done with the genetic DNA fingerprinting, and then we are done with this part of your restriction enzymes, and then we started today our day with the fractionation and centrifugation, then quantitation, and tomorrow we will start with the protein purifications. That's it for today. Thank you very much for being there, and see you tomorrow. Take care. Until then, bye bye. So I've already shared these files with you. So make notes according to it. So it will take some time to get uploaded, but you will definitely get it by today also. So friction enzymes, DNA fingerprinting. Bye bye. See you, students. I have to leave now. You might have another classes, I guess.